Hello everyone, I'm Charlotte from MDPI and I will be your host for today. Welcome to the third live session of the first International Electronic Conference on Biological Diversity, Ecology and Evolution. This conference has been organised by MDPI and the Diversity Journal. I wanted to take this opportunity to thank our chair, Professor Michael Wink, and conference committee for making this conference possible, and also the speakers and the participants for joining us today. The conference is now open for discussion. Until the 31st of March, you can log in with your SciForum account and read and comment all submissions presented in the conference. As you know, live sessions are being organised throughout the conference, so please check the conference website for more information. Registration to the last live session, which is tomorrow, is still open. I would also like to remind everyone that the live session is being recorded and that the recording will be available on the conference's webpage as soon as possible. The webinar is also being live streamed on MDPI's YouTube channel, so hello to those of you watching us on there. Today's session is dedicated to marine and freshwater ecology with a panel of 6 excellent speakers. We will have a time for questions after each talk, so please type your questions in the Q&A box at any time during the session. Please make sure to add the name of the person to whom you're addressing your question. I now hand over the microphone to Professor Wink, who will chair this third session. Thank you, Professor. Enjoy, everyone.
Well, thank you, Charlotte, for the introduction and the opening of the conference. Good afternoon, everybody. Welcome to our third session on marine and freshwater ecology. I will chair this session. I would briefly introduce myself. I'm professor at Heidelberg University. Heidelberg is a city in Germany. You see in the back the famous castle. Maybe you already have seen it. Our university is the oldest university in Germany, founded in 1386, so we are more than 600 years old. My own interest is evolution, systematics, phylogeny, ornithology, and medicinal plants, phytochemistry, and pharmacology and biotechnology. I'm editor-in-chief of Diversity. You know, this is an open access journal founded in 2009. So far, we have published more than 1,250 papers. Our impact factor is 1.4. This is quite good for a journal in ecology and organismic biology. So Diversity has been organizing this conference. So we had 4 sessions, 2 last week, and 2 sessions this week. Our today's session is about marine and freshwater topics. We have 6 speakers. Would remind the speakers the time for each speaker is 20 minutes, including discussion. So with this, no more ado, welcome everybody to our session, and we will start with our first speaker, Daniel Redondo-Gómez. Please open your screen, share your screen with us. So this talk will be on marine and terrestrial scavenging on fish and gull carcasses.
Okay, so hello everyone. Thanks for Professor Wenke for the introduction. First of all, I would like to thank Diversity and MDPI for giving us the opportunity to be here today. And of course, I would like to thank the co-authors and institutions that have made this work possible. And that said, let's get started with a brief introduction, speaking a bit about the recycling of dead organic matter, which is an essential process across ecosystems, as it is fundamental to maintain the stability and functioning of food webs in terrestrial, freshwater, and marine ecosystems. The importance of carrion as a high-quality detritus and scavengers as an important node in the flow of these resources in ecosystem is being increasingly recognized by researchers, but it's true that most studies focus on terrestrial environments, and few studies to date has focused on how scavenging unfolds in marine environments. And most of them deal with deep sea whale falls or the decomposition of pig carcasses for forensic purposes. So there is a knowledge gap in scavenging processes in marine waters, especially in shallow marine waters. The consumption patterns of marine carcasses are different from terrestrial carcasses, maybe for two main reasons. First, the higher mobility of carcasses in the sea, vertical and horizontal mobility due to wave action mainly, and also the longer tissue preservation due to salinity and thermal stability, and also the limited bacterial growth in the sea. As you maybe know, scavengers are facing now a lot of conservation issues due to human activities, and this endanger their populations and jeopardize the ecosystem services they provide. Understanding the differences in carrion cycling among ecosystems and the effect of these human activities on scavenger populations are really emerging needs for conservation now. Here, by simultaneously studying fish and gull carcasses underwater and on land, we aim to identify differences in the scavenging processes between these two environments, focusing especially in the scavenging community structure and also the scavenging efficiency. We conducted our study in Grossa Island. This is a small Mediterranean island located, as you can see here, in the coast of Murcia region in southeastern Spain. As you can see, most of the island is covered by shrub vegetation, and most of the coast is rocky, but there are also some sandy beaches in this side you are seeing now. And around the island, the seabed is dominated by an extensive seagrass meadow of Posidonia oceanica. The scavenging vertebrate fauna on land mainly consists of seabirds, especially yellow-legged gulls. Birds of prey are absent during non-migration periods, with the exception of a resident breeding pair of peregrine falcons. And other potential scavengers in the island, vertebrate scavengers in the island, are Montpellier snakes. Regarding the invertebrate community on land, is dominated by Coleoptera, Diptera, and Hymenoptera. The underwater megafauna is mainly composed of common Mediterranean fish species, like Diplodocus and Oblata species, and other invertebrates, also some invertebrates like octopuses, crabs, or gastropods are also abundant. We conducted this research between July and August 2018 in this island, and we monitored the consumption process of 56 fish and gull carcasses distributed, as you can see here, between the terrestrial and marine environment around the island. And we monitored the carcasses until its complete consumption, or for 7 days if carcasses were not completely consumed. On land, we place one infrared motion trigger camera in front of each carcass to record vertebrate scavengers feeding on it. And to record invertebrate scavengers, we sampled each carcass at four different stages of the experiment. The marine carcasses were monitored, were placed on the seabed at approximately 2 meters depth, and we snorkeled to visit each carcass 5 times per day to record both vertebrate and invertebrate scavengers feeding on each one. Okay, so let's move on to data analysis. And as you see here, we made mainly these three analyses. First, nestedness analysis is an approximation to the structure of the scavenger assemblage on each carcass. A scavenging network is considered nested when the species feeding on carcasses visited by few consumers are a subset of those species feeding on carcasses visited by more consumers. co-occurrence analysis were another approximation to the structure of the community, detecting species appearing in the same carrion more or less frequently than expected by chance. And finally, generalized linear models were carried out to explain the changes in these variables you see here, related to richness, species richness in each carcass, and also consumption and detection times of each carcass. If going to the results of the experiment, the scavenging community consisted in the marine environment of these species you are seeing here, 3 invertebrates and 10 fish species, vertebrates. And the most common scavenger in the marine environment were the gastropods, S.S.S, trunculus, and also the juveniles of the fishes from the Plodus and Oblata species. While in the terrestrial environment, the only vertebrate scavengers was the yellow-legged gull, detected feeding on all fish carcasses and none on the gull carcasses. And 16 species of insects were detected feeding on terrestrial carcasses. The structure of the scavenging network was nested except for fish terrestrial carcasses because it was a particular situation because gulls were detected in all the fish carcasses. And was the only scavenger in these carcasses, but in one carcass we also found some insects. So we found this particular structure you are seeing here. And regarding scavenging efficiency, I want to highlight 2 facts. First, the quick detection by insects in the terrestrial environment, and you can see with this brown line, all carcasses were detected in the moment of its deployment. And it was also remarkable the fast consumption of fish carcasses compared to gull carcasses. And you can see in this dashed line, the difference between fish carcasses and gull carcasses were very, very big. The results of our experiment show clear differences between environments in both the scavenging community and the scavenging efficiency. As you can see here, regarding the scavenging community, the most remarkable difference was the ratio between vertebrate and invertebrate scavengers. As you can see, most species in the marine environment were fishes, were vertebrates, while in the terrestrial environment, most species were vertebrates, and only one vertebrate species, the yellow-legged gull, was detected in the terrestrial environment. And as you can see here, and as I have said before, this community scavenging on fish terrestrial carcasses was the only that were non-nested, and gull carcasses and fish marine carcasses were nested. So this shows a very structured scavenger assemblage and often related to a higher efficiency. But contrary to this general pattern, we found that this community of fish terrestrial carcasses, non-nested and low diverse, was the most efficient consuming these carcasses. This was clearly related with the presence of a very efficient scavenger inhabiting the island, the yellow-legged gull, which quickly monopolized and consumed fish carcasses even before other scavengers, insects, could exploit this resource. And this fact highlights the importance of these carcasses avoided by very, very efficient scavengers to allow other species, insects in our experiment, to complete their life cycle. In our experiment, we found that the scavenging efficiency was higher in the terrestrial environment compared to the marine environment, as previous studies have found. The larger size and high abundance of the yellow-legged gull compared to marine scavengers, and also its high metabolism due to their endothermic nature, are the reason for such greater efficiency. Also, the fast detection by insects in terrestrial environments contribute to this greater efficiency. And however, it also appeared that the consumption time was strongly dependent on the carcass type, even more than the environment, because as you see here, gulls even very often avoided to eat other gulls. So differences between fish and gulls were bigger than between marine and terrestrial environments. Fishes were consumed faster than gulls, as I have said before. And to finish, I wanted to say that this study represents the first experiment with a large sample size comparing the scavenging processes occurring in the same geographical area between marine and terrestrial environments. Such studies, therefore, of fundamental importance for future research on this field. And moreover, the low number of studies conducted on shallow marine water adds value to the results of this experiment. And also given the great differences found between carcass types, we think that further research should focus on underwater consumption patterns of different sources of carrion. And that is all. Thanks again to all the people behind this research, and thank you very much for your attention. If you have any question, please feel free to ask either now or later in the website or by email. Thank you very much.
Well, thank you very much, Daniel, for your interesting talk. So the scavenger system is very exciting and not well studied. So nice work. Maybe I can start the discussion myself, because we did similar experiments several years ago on the mainland terrestrial system. So we're putting out that chicken and look who is going to scavenge. And surprisingly, it was insects again in there first. It was burrowing beetles, Necrophorus, which immediately came within hours and started to work on the carcass. Do these Necrophorus beetles also occur in your environment on the islands, or is it just flies and other things?
Yes, of course, there was a very big community and very diverse community of beetles feeding on the carcasses. But it's true that the first insects feeding on the carcasses used to be flies. Yeah.
But you see, on terrestrial area, it's the burying beetle, which is a specialized beetle for carcasses. Yeah. So you don't have it in Spain.
And yes, I think, I don't know the level of specialization of these beetles on scavenging, but yes, in the island, if I don't remember bad, we have a community of maybe 10 species of beetles feeding on these carcasses. Yeah.
Okay, maybe I can ask another thing for the carcasses underwater. What about squids?
Yeah, we didn't detect any. We didn't detect any squids in these carcasses. Yes, some octopuses, but maybe we recognize that in marine environment our method is not so precise, because we had to snorkel to visit each carcass several times a day. So maybe we are underestimating the number of species feeding on marine carcasses. But yes, we didn't detect any squids feeding on our carcasses.
And for the birds, you only found the gulls as scavengers?
Yeah.
No kites? No kites or vultures?
No, this is a very small island, so it's the vertebrate community is not very diverse,
Okay.
and in some immigration periods you can see some raptors or other potential scavengers around the island, but in our study we did not detect any vertebrate species more than the yellow-legged gull.
Thank you very much.
Thank
Well, is there any questions from the audience?
No, I don't see any questions.
So in this case, thank you again.
Thank you very much.
And we now turn to Poland. Magdalena Grabowska, let's
I
see.
try to connect. Dear Professor, dear Chairman, and dear colleagues, I have a pleasure to present short lecture about springs. We can define a spring as a point at which water flows from an aquifer to the Earth's surface. In my opinion, springs are worth walking because they are among the least recognized aquatic systems. They create some kind of double ecotones because combine aquatic and terrestrial, as well as subterranean and surface water environmental features. They also guarantee high stability of water parameters, for example, temperature, throughout the year. And the most important, remain biodiversity refuges, even in anthropogenically influenced areas. Springs are globally abundant. The algae flora of European springs has been extensively studied for over 200 years. Diatoms are amongst the most common and most abundant organism in springs. Interest of diatom flora in European springs has increased markedly in the late decades, but diatoms from uplands and mountains are more often described, for example, from Alps and Dolomites. My country has also a long tradition in chronobiological studies, dating back 19th century. First studies were examples of first study are manuscripts written, for example, by Gutfiński, Rostafiński, and Liebendans. But algorithmical studies are being continued to this day, among others by Reichel and Wołowski, Kawecka, Żelazna Wieczorek, and Wojta. Spring distribution of springs in Poland is irregular. There are only few areas with spring in the lowlands near city Białystok, Olsztyn, Gdańsk. Therefore, researcher attention is focused on central and southern Poland with uplands, hills, and mountains. Based on results given by Żelazna Wieczorek and Wojtal, we can see that high number of diatom species are presented, many rare or even new for science. For example, Żelazna Wieczorek described 2 new species, Amphora polonica and Navicula fontana, while Wojtal new genus Crenotria and 3 new species. Many classifications are proposed for springs. In my opinion, in hydrobiological and phycological studies, the most useful is typology proposed by Tineman, with 3 types of spring niches. Rheocrene occur where the surge emerges into a defined channel. Helocrenes emerge diffusely in a marshy wet meadow setting rather than having a discrete source. and limnocrene, where groundwater flows from aquifers to one or more pools. My study area is located in lowlands in northeastern Poland. It is a big forest complex protected as Knyszyńska Forest Landscape Park. This forest complex is surrounding my city Białystok. It is the second largest landscape park in my country. Over 200 springs located mostly in river valleys are one of the most precious natural value of Knyszyńska Forest. My study was carried out in 26 springs, mainly in summer, in August 2014 and August 2015. I repeated 2 springs in April 2017. So far, chronological studies of these lowland springs were focused on water quality. Springs represent bicarbonate calcium freshwater habitats. They are mainly saline and alkaline and differ in discharge. The highest discharge is above 50 liters per second. 3 types of spring niches are presented, mostly in forest area. They have stone, gravel, and sand bottom. My diatoms were sampled from stone and sand bottom. The highest discharge was recorded in Rokren in Pieszczaniki. My aim was to study of diatoms diversity in this protected area, recognition of localities of special value. I used light and scanning electron microscopy. I recorded 167 taxa. I didn't find any taxa in 2 springs from unknown reason. And the highest number of taxa was recorded in Limnocren Łaźnie, 36 species. Many diatoms are classified as common taxa. For example, Navicula tripunctata. It tolerates pollution, absent in oligotrophy and acidic waters. Planotidium lanceolatum, recorded from all types of water. And Meridium circulare, forming colony taxa, very common, more common in clean than enriched waters. As fairly common taxa, I described Reimeria sinuata with optimum development in flowing waters of mountains, and Coconeis placentula varietas lineata, widespread taxa with broad range of environmental conditions, but according to Voital, except of saline, carbonated, and sulfide springs. As a rare taxa, I describe it, for example, Flagellariforma nichoides, very rare taxa, usually in low number. I detected it in 2 springs, only single valves. Anomastus tresei, rare in running water. Diploneis crameri, absent from eutrophic water, prefers calcium bicarbonate freshwater habitats. And as valuable taxa, I describe it Coconeis pseudotumensis. It doesn't tolerate even a small amount of pollution. Some taxa are still poorly known. in aspect of, distribution or ecology. For example, Navicula upsaliensis, its distribution is poorly known because it's often confused with two other taxa, Navicula antoni and Navicula minusculus, present in standing and flowing waters, mainly of the lowlands. Amphora eximia, its ecological preferences are not known in detail. But species is frequently is more frequent in undisturbed oligotrophic freshwater habitats, particularly in northern Europe. And one of the largest diatoms, Stauroneis phenictenteron, recorded in one spring, usually from oligosaprobic standing waters. Navicula striolata presents endangered taxa. It has record from 4 others. It has 4 records from Poland. According to Wojtal, species present mainly in upland springs with a sand bottom. It has broad tolerance range for nitrate concentration, but narrow for magnesium and moderate for sodium. Diploneis alpina was first record. It was a first record for Poland. It was recorded in 5 other European countries. It is distinguished in German Red List of plants. Its dimension from lowland springs slightly differ from those proposed by Kramer and Lange-Bartelot. It has slight shorter valves. Based on Polish list of plants and fungi, I distinguished 12 taxa, 1 diatom as endangered taxa, 3 as Vernurep, and 8 as rare taxa. In my opinion, this result confirms the important role of springs in maintaining the biodiversity. Among 167 taxa, I recorded taxa with different environmental requirements, both taxa tolerating narrow and wide range of trophy and pollution, and also preferring different types of waters and landfall. The highest number of species was recorded in typical limnocranic spring, with the longest water retention time. The most common diatoms were partially presented Coconeis placentula varietas lineata and Meridium circulare, and also Diatoma vulgaris, Amphora ovalis. Alongside cosmopolitan and widespread species, many rare species were I have found. For example, Diploneis alpina was first record for Poland. I would like to thank my 2 colleagues, Elżbieta Jekaterynczuk-Rudczyk and Agata Wojtal, for inspiration with springs and diatoms and valuable cooperation. Thank you for your attention.
Well, thank you very much, Magdalena, for your talk about diatoms. It's a completely different world from what I know. Apparently, So my question is, how do you collect your diatoms? Is there a special technique in order to make sure that you get a good sample of everything? Is there a net, or how do you get?
No. I used brushes, and in the case of stone, but in the case of sand, I take surface part of sand and it is difficult to differ sand from valves, but it is impossible, and I prepare slides in special substratum naphrax to microscopy, and I observe empty valves, of course, without protoplasma.
Okay.
After using acids, for example, and perhydrol.
Thank you very much. Any questions from the audience? Maybe a question in general. In many organisms, we now use DNA to identify species. Is this a technique being used with diatoms?
Yes, but I didn't use. I don't use, but it is very important use this technique.
See. Okay, there was a question from the panel, Dr. Nandini.
Yes, Dr. Magdalena, I just wanted to know if the springs are polluted or what, because you found some species which are tolerant to pollution. I thought that the springs are very pristine habitats.
They differ in your water quality. The most is have very good water quality, and diatoms are very good indicators used to assessment, of course, water quality. But some have anthropogenic impact. Yes, mainly in rural area. The most isolated springs have generally have the best water quality, yes, but I need time to compare my species with water chemistry. We have this data because we cooperate with my colleagues, and partially I used these species to receive water quality, and generally the most have very good ecological status, but not all.
Okay. Can I ask another question?
Yes, please.
Thank you. I just wanted to know, how did you determine whether the species are endangered or rare or vulnerable?
Why? How?
How is it done?
My opinion is based on red list of plants. Yes, in many countries are this type of list also for algae, but of course it isn't so easy. Sometimes we still poor know about taxa, and we can think that is endangered, but other study, we should read other study. Sometimes in some places it isn't endangered. Yes, it is important source information. Red list of algae are important source information for me, but not excellent. Yes, in more we have results, we know more. Yes. And
okay, I think
thank you very much.
Thank you.
Yes.
Thank you very much, Dr. Grabowska. We have to move on to our next speaker. It's Dr. Sanja Gottstein. So we are staying in Europe. It's Zagreb now? Croatia?
Yes, I'm from Zagreb, from
Oh,
the
it's good.
Faculty of Science. Yeah. And I would like to present one of my favorite topic, because I'm working on subterranean crustaceans, and my topic is dealing with the arc of crustacean diversity in the cave waters. So my presentation is dealing with this topic. Okay, here we are. So I will jump to this. So it's quite important to say at the beginning that after more than 250 years of research on cave environment, I can say that we are still missing data. Crustaceans, according to the last analysis, form around 43% of the known stable faunal diversity. So I tried to analyze, compare data according to the real analysis, not only perceptions, and the real number of stegobitic, real subterranean crustaceans dealing with only caves, not other habitats like interstitial environment, still missing. So according to this, I will try to present my step of analysis, because I'm focused on the whole world and on the dwelling crustaceans. I try to analyze the species richness first of all, then of course taxonomic diversity and re-evaluation, because during last decades many species, many groups, many genus and species was revised. Then I will present you the analysis according to the biogeographical scale, according to the biogeographical unit of the whole world, and finally the analysis of habitat. So it was the main object of my presentation and my study. Finally, find the knowledge gaps, because thanks to a lot of literature and analysis, even recent literature from the last 2 years, I found many gaps, especially according to the classification and analysis of subterranean dealing with crustaceans as a main object. So the whole assessment, my assessment of crustacean diversity in caves was passed through different phases. First of all, it was a sort of my passion, I must say. First was to form a appropriate taxonomic database. I start with the Excel. Then I study more than 2,000 published papers after the main literature source, which is Fauna Mundi of Gottstein from 1996, and other literature is like Encyclopedia Biospeleologica in 3 volumes and Encyclopedia of Caves from 2009, which is the 3rd edition of the great book. So more than this, I also use various sources from the web, like World Register of Marine Species, but there are missing data on the real habitat for the crustaceans. So only few of animals, few of species actually have those data. Then I then crustacean cave species adequately, which is described by the end of December 2020. And also I analyzed a lot of so-called gray literature. So gray literature was sole focus of my analysis. So, new taxa, new combination of crustaceans in the cave environment. There are many and according to the first data, well, I can see that there are still many work to do, but the total number of cave stigopitid crustaceans is presented now according on the class level, which we have more than 100 species belongs to the Malacostraca, which is dominant in environment. Then the next one is Stilopoda with more than 1,000, ostracods with more than 600, then and brachiopods. So I can say that it's 3,100 species and subspecies, and it is the first type of this type of analysis in the world. So, 10, I can say that I enlarged the number of crustaceans times. The total number of Cryptic as the dominant taxa, well, with the highest number of species among amphipods. So I counted more than 7 species, which is obligate cave crustaceans, which are not in other subterranean environments, and then paired others, isopods, decapods, and all other is with less than 50. So total paired with 1,473 species. More interesting group with number of species is amphipods, my favorite group, which I analyzed in a detail. So first of all, I analyzed the appearance of number of genera and of course number of species. I found first of all, I found of course the highest number of species and subspecies is in the family Niphargidae, dominant taxa among amphipods. Then the highest number of genera was among family Hagiidae. So it was 26 genera and 116 species and subspecies of Niphargidae. So altogether, I recorded 38 families, which is higher than previous recent analysis, which was less than this. Even there analyzed all stigopitid crustaceans, and so I recorded families which is monotypic, quite interesting according to this analogy, like Algae, Niphargidae, Cotomsaridae, and Seborgidae. So, Olin, I can say that according to the geographical scale, I analyzed of the region according to the appearance of cave crustaceans, and I found the dominant regions is Palearctic, almost 50% of all crustaceans with 49%. And after that appeared Nearctic regions with USA, of course, Neotropical with 50%, and then there's Australasia and regions, so India, China, and all others. Then regions with only 2% of the obligate cave crustaceans and oriental regions, which extended on this area. So Hawaii and Galapagos is also included in this analysis. So the cave Walsingham is a hotspot of crustaceans, which we can see according this even on the highest level, not on the species level, on the level with more than 40 stegobounds there. So there are many spots where we can find species. But to habitat type analysis, crustaceans is appeared in only caves but in lava tubes too, and other type of caves. So dominant caves according to my analysis, and according to the analysis of the type, the dominant is freshwater, and then appeared fish and marine with the same quantity. So in total, 79 living in freshwater. If we analyze and compare the world distribution of karstic rocks, we can see that in, for example, in Africa, we have a lot of karstic regions which are not still analyzed and studied. So especially, for example, southern part of Australia. So we are still missing the data according to the new analysis. For example, in Europe and especially in Dinaric karst, which is one of the most diverse region according to the caves and cave animals and crustaceans too. We are still missing many data. For example, in Croatia, we are still missing data on the deepest cave. For example, one which is more than 1,040 meters depth. Even we reach the aquifer, and there are many of them is in the Dinarics because we have an opportunity to research them. So we are still missing the data on cave crustaceans in those deep environments. And I can say, according to my analysis, what we can conclude that according to the analysis on the level of species and the level of taxonomic diversity, well, we are still missing the data from some old literature and analysis of literature, especially on geographical scale, because in some old description of the animals, we are missing the data where it really appeared, and especially according to the ecology. So we are missing the data where it was found. We have a description of the animals, but we haven't actually the data. Is it in the cave environment or in? So there are many still. So thank you for the attention, and I'm welcome for the question.
Well, thank you very much, Sanja Gottstein, for your talk about this cryptic biodiversity in cave. Is there any question from the audience? Well, as you already pointed out, apparently there are some areas in the world where people have been studying crustaceans in caves, and in many other areas in the world, people have not been looking into caves. Is that true?
True. Of course, I can say that last 2 or 3 decades, the interest was raised, but previously it was very hard to reach the cave environment. Even I can say that Emil Rakovica who was one of the first biospeleologists from Romania, researched caves even at the beginning of 20th century very well, and he occupied the whole Europe to research those environments. But the data are still missing on crustaceans. Yeah, so and we don't know exact how many of cave animals. We have the data on speculations. We have a data on the whole stegobitic crustaceans, but what of crustaceans is real adapted to the cave environment we are still missing.
Okay, I see. And also from the last picture, I realized it's really an adventure to go into caves.
Yeah.
So you also have to dive. Yeah.
Yes. Yeah. The specialization for scuba diving in cave environment, thanks to Thomas Iliffe from USA, I can say that the data from this environment is higher and higher, especially from Yucatan Peninsula, from Mexico. So we can, thanks to those explorers which are exciting on this environment, which is enthusiast. And also I can say that all those data which are made in my presentations is thanks to enthusiasm during last 5 years, I can say, but mostly my husband and me, because he helped me to collect the data from the literature, from the websites, to analyze, to find where is really crustaceans of cave environment appeared. Yeah.
Well, thank you very much. I think we have to move on with the program. So that was an interesting expedition into caves.
Yeah, thank you for the opportunity to be here.
Thank you, Ms. Gottstein. So now we move to Fernando Fernández-Alvarez about cryptic biodiversity, a common phenomenon among Atlantic Ocean squids. Fernando.
Yes. Hello. The title of my presentation is Cryptic Biodiversity, a common phenomenon among Atlantic Oceanic squids? And I am presenting it in behalf of myself and my co-authors, Ruye Villanueva and Luis Alcock. So the first thing regarding oceanic squids distribution ranges is that they usually have huge distribution ranges. Most of the species are described as having a cosmopolitan distribution range, and in some cases, like in this one, it's very interesting because this cosmopolitan distribution range is not continuous. For instance, in the case of the neon flying squid, the one of the photo, North Pacific populations are not in contact with the South Pacific populations, and the same happens in the Atlantic because it has an antitropical cosmopolitan distribution. And this is very strange in assuming conspecificity. So this one was one of the first examples we wanted to test. And in this work, we used several samples around the world of these putative only species of neon flying squid, and we found out that in reality it were 4 different species, and the North Atlantic and Mediterranean one was different than the one in the South Atlantic and the Indian Ocean, and the same happened in the Pacific. So in this case, it was some cryptic biodiversity. And when we look at the distribution ranges of other species, we found similar cases. For instance, we have these 3 species with huge distribution ranges, and there are some of the ones that I test in this work. So the objective of this work is to test if in Atlantic oceanic squids we have cryptic biodiversity. And for that, we had almost 200 samples of 12 different oceanic squids coming from different oceanic cruises. For instance, we have the Mafia cruise from Brazil to the Canary Islands, and then we have the Batipelagic from Morocco to Iceland, and also this one, the Summer from Barcelona to Vigo. So many of these species cross at least one of these oceanic barriers I show in this picture, like the Canary Current, the Atlantic equatorial currents, and others like the Strait of Gibraltar, the Almeria Oran Front, etc. So these 12 species belong to seven different families of oceanic squids, and they have different life stages. Some of them are epipelagic, for instance, this Pterygioteutes gemmata and Pyroteutis margaritifera, and other ones are more live at deeper waters, Science and Cystocheurus lesseuri and most of the other ones. For testing this diversity, we collect the squids, and after a dissection, we collect a small tissue snap that was fixed in ethanol for molecular purposes, and the remaining part of the body was fixed in formalin, and it was stored in a scientific institution, so anyone in the world can check it out, the identification of these samples. With the tissue in ethanol, we perform a DNA extraction, and then we perform a PCR amplification using the Folmer's region of the mitochondrial gene CO1. That is the common marker used in DNA in DNA barcoding of animals. We sequence these amplicons, and we obtain the sequences for this marker, and the matrix was complemented with sequences coming from the online database GenBank. With this matrix, we perform phylogenetic analysis, like maximum likelihood or coalescence analysis, and the output of this analysis was used to perform molecular species delimitation methods, like these two ones, the BPTP and the JMYC, that are commonly used for test if there are several species within a name. And then we also perform distance analysis with the sequences. So we test if the distances among the specimens within a clade were smaller than between clades, and so on. I will now, in the results session, I will only comment the results of the species that showed some kind of cryptic biodiversity, and you will see this map depicting all the samples we had, and these colors of these dots match the ones in the cladogram. So this red clade corresponds for all these red dots in the map. And at the right of the cladogram, you also have the results of the three species delimitation methods, the distance analysis, the BPTP, and the JMNC. And in this case, in Tenopteris sicula, we download all the sequence from GenBank as well as source samples, and some of them were not identified, like this Tenopteris care 2, and other ones. And we also have this other one that was Tenopteris canariensis, that is a different species. But coming back to Tenopteris sicula, that is the species we were testing here, we found 4 cryptic lineages, so different species that were identified as Tenopteris sicula, and 2 of them came from the Atlantic, that are the ones in red, and in orange, and 2 others came from the Pacific, that are the one in yellow and the violet clade. The one in red, the red samples, are the nominal species, because Tenopteris sicula was described in the Mediterranean, and the remaining 3 species identified as Tenopteris sicula are new species, or some species that has a name that needs to be resurrected. Moving for the family Ancystrocheiridae, this family is formed by a single species, so it's a monotypic family, and this species is Ancystrocheurus lesseuri. We used sequences from Yimban, from Pacific waters, and then also we add our Atlantic specimens and some other sequence from the Gulf of Mexico. And we found 3 different species. The species delimitation methods were more or less concordant, but the PTP analysis found 3 different species within the Pacific one. This is likely an artifact. So we can say that we have 3 different species among Ancystrocheirus, the species Ancystrocheirus lesseuri. But we prefer to maintain all these species as unidentified because the type locality of Ancystrocheirus lesseuri, the type species of the genus, came from the Indo-Pacific, and we don't have any sequence from there, so we prefer to keep the species as unidentified. Then we have the genus Abraliopsis, that is a complicated one, because all our samples from the Atlantic waters were identified as Abraliopsis morrisi. In the Atlantic, there were only 2 previously described species of Abraliopsis, in North Atlantic, that were Abraliopsis atlantica, that are identified here as this brown dot, and then Abraliopsis morrisi. Or some of our sequences, the ones in orange, match the ones in Yimban for Abraliopsis morrisi. And then we have this parallargy that didn't match anything from Yimban, and then 3 samples from this equatorial area in the Atlantic that didn't match any other sequences as well. So these 2 species can be 2 species that has been described for the South Atlantic, like Abraliopsis hills-christi, or maybe this Abraliopsis species Sita, that is one that is not described yet, but it can be that. So either we have 2 new records of species from the South Atlantic, or we find new species of Abraliopsis. In the case of Galiteutis armata, we have several specimens from North Atlantic identified as this species, and also from the Mediterranean. And in the Atlantic waters and Mediterranean, there is only a single species described, that is Galiteutis armata. But our analysis found that these sequences from South Atlantic, from Yimban, were a different species, and also we have 2 different species among our samples. So there is also cryptic biodiversity within Galiteutis armata in the Atlantic waters. And the most interesting case is Helicocrantia pfefferi, that is also a cosmopolitan species, that we found 5 different species. And I will comment that in more depth in the next slide. So you can have here this map that besides these sample points, we also have here the main currents of all the oceans. And as you can see here, we have depicted the dots, so we can use these currents to have some hypotheses regarding their speciation. In the case, the first 2 species we found are these red and orange dots that has a low divergence values. They have 2% of divergence between them. That is a very low value between different species in oceanic squids. Then we have this yellow sequence from South Atlantic that is also a different species that has 3% of divergence between the other 2 species. And as you can see here, these red dots can be isolated from the orange ones by the Canary Current at the North Equatorial Current, And then we have this yellow dot that is isolated maybe by this subequatorial Atlantic current and the Brazil current as well. So maybe these are an incipient speciation phenomena that it's triggered by these currents and the isolation that it produces in these squids. Then we also have this violet dot that is another North Atlantic species that has a higher level of divergence. You have here 12% to 9% of divergence in comparison with the other species I just mentioned. And then we have the last one, the last Atlantic species, that is this white one that has a larger distance with the remaining species that came from 14% to 17%. And the distance among each of these clades, it's far less important. We have species that are almost equal, and then we have this moderate level of divergence in the violet sequences that came from 0 to 0.6%. So there is a gap between the intraclade and the interclade distances that is a good estimation about the effectiveness of this molecular marker to discover cryptic biodiversity. So as a summary of this complicated assemblage of different species, we have 3 Atlantic species with low levels of divergence between them, and then we have 2 other species that are more distantly related in Atlantic waters. And we don't have anything from the Pacific to check it out. So these species can be there as well. And then we have this Pterygiodontis gemmata that is, in this particular case, we didn't find any signal of cryptic biodiversity within Atlantic waters. But if we compare these Atlantic sequences with the sequences from New Zealand, from GenBank, we found out that all the 3 species delimitation analysis found that there are different species, and they didn't even form a monophyletic clade. As you can see here, the New Zealand species, Pterygiodontis CF gemmata, it's more related with Pterygiodontis giardi than with Atlantic Pterygiodontis gemmata. So as conclusions of this talk, we can say that we found almost 50, we found cryptic biodiversity in 50% of the species tested. So we found in 6 out of 12 tested species. The number of cryptic lineages range from 2 in the case of Tenopteris scula and Ascistocheurus species to 3 in the case of Galitotis armata and 5 in Helicocranchia pfefferi. It is not clear how many lineages, new lineages, are within Abraliopsis, but at least we have 2 species in the Atlantic that can be new records or new species. The divergence among cryptic lineages of individuals of the same nominal species range from 2 to 70%, so they likely represent different stages of divergence between such putative speciation phenomena. The molecular species delimitation methods were generally consistent and useful for the discover cryptic biodiversity among Atlantic Oceanic squids. The Canary and the Atlantic equatorial currents can be responsible of some of these cases of population isolation and subsequent speciation phenomena, but we need more study using larger number of specimens to test these hypotheses. And it is necessary to develop more studies studying the biodiversity at the morphological and molecular level of these animals, the oceanic squids, in my opinion, at a global scale. So thank you so much for your attention. Thank you for organizing this interesting conference. And if anyone wants to contact me, we can do it through my email, my Twitter handle, or my ResearchGate profile. And also thank you to my funders. And if you have any questions, I will be happy to answer it.
Well, thank you very much, Fernando, for your nice talk. An interesting topic of this cryptic biodiversity in oceans. Well, maybe you can start with one question. You only, as far as you mentioned, you only looked at the CO1,
Yeah.
which is a nice marker. But normally one goes beyond the CO1, taking other mitochondrial genes and even more importantly, some nuclear genes. Have you done this, or are you planning to do this?
We are in the process of doing that. Our idea, we are now performing a shallow whole genome sequencing method that is known as genome skimming, that allows you to retrieve the complete mitogenome of these animals, and also they allow you to obtain the complete ribosomal nuclear cluster. So the idea is to develop ITS markers using this complete ribosomal cluster and use that for checking out with ITS, and also check out the full mitogenome of this species, testing the oceanic barriers. That means the ones that we found cryptic biodiversity in populations in different parts of our oceanic barrier.
Excellent. Because, well, if you use only mitochondrial DNA, you probably only have maternal lines.
Yeah.
And you cannot detect hybridization.
Right.
So my question is, does hybridization occur in squids? I have no idea. So I'm not a marine biologist. So because in most animals we have hybridization, and you can only detect it if you look into nuclear genes.
Yeah, right.
What's the situation here?
Yeah, the thing is, as far as I know, there is no record of hybridization between these animals. But as far as I know, there were no one trying to check that. So I didn't find any example in the literature about that. So I think it's unlikely, but it can be one of the things that can happen. Yeah.
Yes, as soon as you do NGS, the genome studies, you will recognize what's going on.
Right. Yeah.
Excellent. Any more questions in the panel? As time is already going on, so I proceed now. Thank you again.
Thank you very much.
Muchas gracias. And we now move to our last two speakers coming from Mexico. So it's early morning there. Buenos dias to Mexico. So our first talk is by Professor Sarma on Rotifer diversity.
Thank you very much. And then I will take you back to Mexico. Now it's morning here at 8:24. In this presentation, I would like to make some comments and observations on the Rotifer diversity in Mexico. This is a group that I have been working for the last 35 years or a little bit more. And first of all, I would like to tell you that if you make collect 1 liter of water from any given water body, you find 3 major groups of zooplankton, ciliates, rotifer, and crustaceans consisting of cladocerans and copepods. How do we understand the rotifers? These are the pseudocoelomate bilaterians. They have a great relation with Acanthocephala using the molecular tools. Morphologically, they are completely different because all Acanthocephala are 100% parasites. There are very few rotifers are actually parasites. Fortunately, you do not have to come to Mexico to study the rotifers. They are present everywhere. Wherever there's an aquatic or semi-aquatic environment in the world, you find them. They're both marine and freshwater, although they are more common in the freshwater. Structurally, you can identify the rotifers without microscope. You cannot identify because most rotifers are less than 1 millimeter. That is, I cannot detect. So you need a microscope. The anterior part of the animal called ciliated corona, it is useful for swimming and to collect the food particles. One of the important features of the rotifers is the presence of mastocs of the trophi, which is used to cut and hold the prey and cut the prey into or algae into smaller bits. Another feature which also shares with the nematodes is the constant number of nuclei and cells. We use here the term nuclei because the animals, all the organs of rotifers are in the syncytial state. That is, the plasma membrane between the cells is vanished. So you can only detect the number of cells on the basis of the number of nuclei that is easy to count. Because of the consistency of the number of nuclei or the number of cells, each rotifer on average about 1000 cells, they have no capacity of regeneration. In the moment the animal is born, the number of cells that the animal should have are already predetermined. There will be no more post hatching cell division. So the growth is only accumulation of organic material in the body in each cell. So that is why the adult and the neonate size is just twice. Adults are twice the size of the neonate because there is no cell division anymore. Because they are primitive animals, there is no respiratory and circulatory system, and most rotifers reproduce by parthenogenesis. And what is important for us to study? Well, they are very important link in the aquatic food chain, especially in the plankton. So they feed on algae, they feed on detritus, they feed on bacteria. Some of the carnivorous rotifers also feed on ciliates. And the energy trapped there is transferred to the next higher level, like fish larvae or other carnivorous plankton. So they are sensitive to changes in the water quality. Small change in the pH, temperature, salinity, and dissolved oxygen level, or even carbonate, bicarbonate levels, the certain species disappear, resistant species appear, or even changes in the population level and composition of the zooplankton. The rotifer zooplankton will also change with the small changes in the water quality. They are the live food in aquaculture. Almost any fish larva, both edible and ornamental species, they feed on the rotifers. And because they're sensitive, they are also used in evaluating the industrial effluents in the ecotoxicological observations. Although many people believe that rotifers and Acanthocephala should be combined into one phylum, still there is not any remarkable agreement among them. The present accepted classification for rotifer, there are two classes, class Pararotatoria and class Aerorotatoria. Class Pararotatoria has only one genus and 4 species, and most of them are associated as epizoic on the Nebalia and the Crustacean, and Nebalia has been reported from Mexico, but the species, the genus Sison has not been recorded so far, but it is not very far away that the rotiferologists also study on this epizoic species. Eurotatoria is very important. There are two subclass Diloidea and subclass Monogononta. Subclass Diloidea is very important from the point of evolution. There is no sexual reproduction. 100% males are absent. All the populations are, there is no male at all the population consisting of 100% females. Yet there is a high species diversity. Then comes the Monogononta. There are about 1600 species all over the world. And rotifers are usually identified by their trophi. And the trophi consisting of 7 different parts, and each of them can be modified depending on the food and feeding habits of the rotifer. There are in all 9 different types of trophi. If you see the history of the rotifer research in Mexico, until the 1990s is mostly 1960s, started in 1901, the description of an Lacana species, and until mid-90s, the studies mostly are on the limnological side, description of 1 or 2 genus and mentioning of the rotifers as a group, but not a very serious systematic study on the rotifers. In the second phase until 90s, you have descriptions of new records, comparison of the species of rotifers from Mexico with other countries and so on. Now we are in the third phase where we have both molecular and morphological descriptions of the species and description of many new cryptic species. We are in the exponential phase since the 1990s, since the mid-90s to until now. We hope it will continue. The Mexican rotifer, Mexico is considered to be one of the 12 mega diverse nations. We have more than 100,000, 120,000 species of both plants and animals. So it is considered mega diverse. And the governmental agency responsible for maintaining the inventory of the species is called CONABIO, the National Commission for Knowledge and Use of Biodiversity. They maintain the records of the species known from Mexico. And here you have an example. They also mentioned about the Rotifer. There are, in the world level, the 1,800, the information is a little bit about 20 years old, and it requires upgradation. At least 20 years ago, we have 300 and more species of rotifers. But why do we have a very high number? Well, there is a history that we have about 70 very large lakes of 10,000 hectares of area and 14,000 reservoirs, and we have also more than 200 rivers, and more than 25 of them are more than 250 kilometers long. We also have the rivers, transnational rivers here, which are mentioned below here, like Rio Bravo, and some important lakes like Chapala, Pascora. These are also the touristic places that generate a lot of revenue for the government. And what is the current status of rotifer research all over the world and also in Mexico in terms of the species diversity, species richness? I would like to say that there are 5 orders, 4 of them are recorded already for Mexico, and then we are very happy 80% of the contributions are from, at least we meet with 80% of the rotifer orders. And when it comes to the family, there are 34 families, 27 of them are already recorded, and we have also again a satisfaction of 80% of the contribution from Mexico. At least we have a representation. And in terms of the genera, there are 130 in the world and 71 of them are already recorded. And the number of species, 2,150 species, and of which we have only 400 species. We are 20% 20% of the species, but still higher in terms of global diversity of all organisms. Mexico holds 10 to 12% of the species of the world. That is why it is considered to be one of the mega diverse nations. And what is the probability that we get a certain number of species in a given water body? Comes vary depending on the different regions of the country. So central Mexican region usually offers on an average about 50 to 60 species of rotifers. These are all one-time collections. You can go at any time in the year and then make a sample with 50 microns mesh and filter between 50 to 100 liters and then concentrate and observe on the microscope. You do not have to check the live specimens just to have an idea. Then you get about 50 to 60 species. When you come to this southeast Mexico, the number is slightly lower because very few water bodies are studied. Its number is increasing. This we have about 40 species. But if you come to the northern region of Mexico, we have desert regions. There are variability. There is a high variability of the species occurrences depending on the year of collection, and if the dry period you don't get much because you need to take the mud and then try to hatch them, and the number can be variable. And but these are all one. So what will happen if we make collections throughout the year? So here we have some information on more than a dozen water bodies that we have been studying for the last 25 years, and full year collections in some areas for some water bodies we have been collecting for the last 10 years or so. So the average, the number is much higher. So compared to one-time collections, seasonal collections always yield higher number of the species richness. So what are the, out of the 400 genera, 400 species that we have, most of them are represented only by 5 genera. These 5 genera are common, independent of the water body that you collect in Mexico. There is the genera Lecane, Trichocerca, Brachynocephalodella, and Lepidella. Each of them has on an average 20 species recorded. The most diverse, of course, is the Lecane. In the world, there are 120 species, and 70 of them are already known from Mexico. They are very beautiful to observe. You do not need a live sample, and then fixed samples are sufficient to know the diversity. And Brachyurus usually is found in the eutrophic waters. Many water bodies in Mexico are eutrophic, and they are indicators of water quality. Usually they are abundant in the eutrophic waters, and the most common are Brachyurus calciflorus. These are all cosmopolitan species. And one of the most important species, most common also in Mexico, is Brachyurus hawaiiensis. It is found abundantly in all the water bodies. Then we also have Brachyons rubens, Brachyons bidentatus, quadridentatus, and Brachyons picatulus is a species complex. It's not easy to identify the species just based on the morphology. You need molecular tools. Then we also have some very rare and interesting species. For example, you have Brachynocyte rhodinoides in Lake Chapala, and it shows a lot of morphological variations, and then probably molecular tools are necessary to decipher whether whether it is a species complex or it is the same morphological variation. And Brachyons havanensis is so abundant and then so proliferate in its reproductive rate is very high, and therefore it is considered to be invasive in some countries in Europe, especially because it can dislodge local zooplankton species. Then another group, another genus, which is also common, is a Trichocerca. The relation between Brachyons and Trichocerca is usually used as indicators of water quality. And Trichocerca is usually present in the oligotrophic conditions. Some of the most common species you find in central Mexico is a Trichocerca similis, Trichocerca elongata, pustula, Trichocerca cylindrica, Trichocerca stylata, among many others. And then comes the complicated genus Lepardella. Is common in both shallow and deep water lakes, and the diversity is high, but the body size very small, usually between 50 to 80 micrometer, and identifications are a bit complicated, but again, you do not need a live sample. Formalin preserved samples are sufficient to identify at species level. Cephalodella is the genus among all the Rotifer genera. This genus is most diverse with more than 200 species, and morphologically is very complex because the trophy is obligatory to study. At times, because of their minute size, they can reach the densities more than 1,500 individuals per liter. 1 milliliter, put 1 milliliter take from a given water body in certain months of the year, you find at least one individual of this genus. Then comes to the Cephalodella. Then comes the sessile rotifers. These sessile rotifers cannot be identified without live individuals, and they are usually associated with the plankton, with the plants, macrophytes. All over the world, there are about 100 species, and 35 of them are already known. And many more complications for identification of these species, especially there are variations in the at the subspecific, infraspecific level. Then Keratella is common, Keratella mexicana. You find here is you find that morphologically is distinct, is an endemic species to Mexico. Then if you compare the Mexican rotifer diversity with that of neighboring countries sharing border with Mexico, that the United States has 600 plus, about 620 or so on, and the number is still not confirmed. And then Mexico has 400 species. If you compare with the South American, Central South American species, some of the smaller nations have got. So Mexico is considered to be a transition area between South and North America. Therefore, we expect the number is high. Therefore, Mexico also has endemic species. Here is an example. And there are about half a dozen endemic species well defined morphologically. So the diversity of special rotifers, as already mentioned, then comes to the Mexico deserves, because of its unique position between the South and North America, and it has potential at least 500 species and more. And here is an extrapolation curve suggests we have 500 species, but the main problem is that we have no reliable data from the 9 out of 31 plus Mexico City federal entities. 9 of them we have no reliable information. That is why we are not able to reach the 500 plus species. And brackish water rotifer species are again did not receive much attention. That's why we have no much reliable information. Third is a third aspect is the deloid rotifers. These are for last 80 million years, there are no males, they are not hermaphrodites, and there is no meiosis. Yet the diversity is 500 species all over the world, and people who work on genetics consider it as an evolutionary scandal. And we also have saline lakes, and some of the deep saline lakes have very low diversity. However, temporary saline lakes have high diversity. And molecular taxonomy and cryptic species started only recently, about 10-15 years ago. And then some of the genera like Plankton, Brachans, Platias, Keratella, Mytilena, Testudella, they have about cryptic species equivalent to 50 taxa. So, however, molecular taxa is based on morphological taxa is based on molecular studies, especially are not based on the culture taxa. We need to culture them because some of them may have in gut another other species. And what are the problems that we have currently? Up to 31 states and the federal entity of Mexico City, 9 states have information. Both point and seasonal corrections are needed. Taxonomical studies are well carried out in only few states. Diversity of Mexican delights is not known fully, and endemism of course is present, and cryptic species need to be formally described. They are identified, but not they are formally described. And I thank BD, and I thank you for listening to my talk.
Well, muchas gracias. That was a very nice summary of rotifers in Mexico. Well, obviously a difficult topic and not known to many zoologists. It's also a cryptic world. And as you mentioned, probably there are much more species around once you go to the molecular level. Thank you very much. One question, you have a number of caves in Mexico. Are the rotifers also going into caves?
Yes, we do. But I have to warn you because for cave collection, which previous speaker mentioned about it, so one of our colleagues died because caves also harbor many unknown or even dangerous fungus. So if you do not have proper equipment and if you go into the caves, and then this one of my colleagues died because this fungus penetrated into the respiratory system and spread fungal hyphae throughout the system of respiration, and she died. So it is important that before you go, you are fully equipped.
Okay.
But for many cave fauna, for cladocerans and copepods are well described.
Okay. I have a question also for the distribution of rotifers in your country. Are they distributed by birds, by water birds, from one lake to another? So
By many
feed and
yes, there are many means. Resting eggs are very light and they can be blown away by winds, especially the desert winds. So they cross. They do not need visa to go to US, and then they just cross frequently. And many of the desert species from Mexico are also found in Texas, for example. Number one, the second is anthropogenic, and the third is also by birds, because they feed on them and the resting eggs are caught on the web on their legs, and then they transfer between. And finally, the distribution of rotifers is also dependent on the rotiferologists.
yes.
Rotifer workers. So wherever there are rotifer workers, there is more diversity of rotifers.
That's normally in zoology. If you have specialists, you have more species. Well, a final question. How do your rotifers survive difficult times? So when the water body gets dried up, or in the north when it's frozen, how do they survive? Do they have stages?
Yes.
Yes.
Okay. The rotifers survive in two means. One, the adults, in case of delights, adult can be dried at any stage and develop into a kind of encystment. They're called cryptobiosis, and they live for many years, 40 to 50 years, they live without any problem. And monogonal rotifers reproduce sexually when the conditions are not favorable, and these resting eggs can remain there for up to 100 years. And in case of Artemia, the cyst can last for 1,000 years.
Okay, very interesting. Well, any questions from the panel or the audience? It doesn't look like. So also by time, thank you again. Muchas gracias. We stay within your area. Now we go to exotic plankton species in Mexican freshwaters. And now the floor is yours, Dr. Nandini. Well, I think we come to the end of our afternoon session on marine organisms. So we heard several talks from diatoms to rotifers to introduce species. We heard about squids, we heard about crustaceans in caves, we heard about carcasses in the sea and all the problems. So we had a very diverse topics, people coming from different parts of the world, young people, established researchers. I would like to thank all of you for your contributions. Well, it's a challenge these days with Corona around, so we have to go for Zoom conferences. But I personally think it also has some advantages. Now we can participate in conferences without being able to travel. So that's also nice, and to be connected with the world of scientists. Is there any comment from the panel at this stage? Seems not to be the case. So it's now my part to say thank you again and hand over to Charlotte.
Thank you, Professor Wink. Yes, I would just like to thank you for chairing this session and all the speakers today for taking part, as well as the attendees. I'd like to remind everyone that the last session will be held tomorrow at 9 o'clock in the morning European time. So if you haven't yet registered, you can register on the conference website. I'm wishing everyone a pleasant day, afternoon, evening, wherever you are in the world. And I hope to see you all tomorrow. Bye-bye.
Thank you very much. Bye-bye.
Bye-bye. See you tomorrow. Bye-bye.
Bye-bye.
Okay, bye.